in this video, we will discuss about techniques used in cell biology or techniques in cell biology. Technique can be defined. Method of doing something very expertly is known as techniques. Various techniques have been useful for isolating various cell components. Of these, differential staining, centrifugation, tissue culture, chromatography, electrophoresis are the most common, and microscopy are the most common techniques. As I have told that techniques can be defined as skillful or efficient way of doing something. First of all, the first techniques which are used in cell biology is microscopy. First of all, micro means small and scope means to view, to look, to vision. It is an optical instrument with the help of which we can see those things which cannot be seen with our naked eyes. As an optical instrument for viewing very small object, typically made by magnifying several hundred times. Each and every microscope, there are various types of microscope. For example, simple microscope, electron microscope, transmission electron microscope, and scanning electron microscope. These different types of microscope, it depends upon the magnification as well as the resolution power. Then what is magnification power? Magnification is basically the capacity of microscope to enlarge the apparent size of the object is known as magnification, or the ability of microscope to increase the apparent size of the object. It is denoted by the word or the suffix x. The magnification power of light microscope is 10,000 times, while those of transmission electron microscope, it is 10 lakhs times or 1 million times. Now, what is resolution? Resolution can be defined. It is the ability of microscope to distinguish or separate as distinct subjects that lie in close proximity. Or we can also be defined the resolution power is the ability to distinguish close object as being separate from one another, or it is the ability of microscope to distinguish between the two lines points in an object, or ability to clearly separate two points situated close together. The resolution power of light microscope is 250 nanometer, while that of transmission electron microscope it have 0.2 nanometer resolving power. In this picture, you see the compound microscope. This is the eyepiece of the microscope. It have a lens, and this is known as body tube of the compound microscope. This screw is known as coarse adjustment screw. This is known as fine adjustment screw. It is known as arm of the microscope. It is known as the resolving nose piece, and these are various types of objective. And these here it is known as cover slip. It is known as condenser. Here it is mirror. This is the base, and it is known as the inclination joints. The eyepiece lens as well as the objective lens, they are multiplied, and the magnification power of microscope can be obtained. So this is ordinary simple compound microscope used commonly in laboratory. And you see in this picture, it is the electron microscope. There are, as I have told, that there are various classification of microscope. Simple microscope, which is simply known as magnifying glass. Compound microscope, as you have seen in the diagrams. And then the electron microscope, which have two types, scanning electron microscope as well as transmission electron microscope. Now we will go to another techniques which is used in cell biology or cytology, is known as staining. Staining can be defined. It is a microscopic technique used to enhance the clarity of microscopic image. As you know that most biological structures are transparent, so staining techniques are used. A technique used to produce a contrast to different structure, tissue, staining evident cell and tissue component, and also permit distinction to be made between them. There are various classification of stain. Some are temporary stains in which the color from the tissues or from the object can be easily withdrawn, while permanent stain is a stain with the help of which when we color any biological tissue which are transparent, the color is permanently present for long time, which is known as permanent stain. Then another classification, that is what is vital stain. Vital stain, those stains that cannot harm living tissues are called vital stains. Or the stain which is used in low concentration and non-toxic to living tissues, that for example methylene blue or neutral red is the example of a vital stain. Then what is counter stain? A counter stain is a stain with color contrasting to the principal stain, making the stain structure easily visible using a microscope. Or a counter stain is a stain with color contrasting to the principal stain, making the stain structure easily visible using a microscope. So counter stain is used in differential staining, Isen counter stain to hematoxylin, crystal violet endgame staining. In this diagram, you can see that various types of stain: aniline blue, borax, carmine, iosine, fulgen stain, leishman stain, methylene blue, safranine, aniline sulfate, iodine solution, Scholl solution. These are various types of permanent as well as temporary stains in which their final color is different. For example, in this diagram, you can see that safranine is the name of a stain in which their final color is a red or a purple when we apply it on a transparent biological tissues. If you like the video, please share, subscribe and watch. Thank you very much.